Good afternoon, everyone. Welcome back to Virology. We've talked a lot about how viruses change this semester, and today we're going to talk about the mechanisms behind that. We're going to talk about evolution. As Alice said, around here it takes all the running you can do just to stay in the same place. We'll get to that, though. There is a thing in biology called the Red Queen hypothesis. Have you heard of it? What are they teaching you here at Columbia? Red Queen hypothesis is a big deal. It just means you're going back and forth because, that's what the Queen said. Around here, all the running you could do just to stay in the same place. That's the Red Queen hypothesis. And we're going to illustrate it today in terms of viruses and their hosts. But first, evolution. So we all think of Charles Darwin as putting forth a theory of evolution, which is no longer a theory. It happens. Evolution is real. And what he did is he made observations of many different organisms and noted how they were related to each other. And on the left is one of his notebooks where he wrote, "I think," and then he drew a phylogenetic tree, understanding how all these organisms were related to some ancestor. And once, years ago at a virology meeting, I saw someone with that tattooed on their arm there. Isn't that cool? I think. And the phylogenetic tree. You have to really like Darwin to put that on, or science in general. That is how organisms are related to each other. They evolved from an ancestor, and we're going to talk about that today in viruses. So here's a relatively simple scheme of evolution. We're going to talk about it in terms of a host protein, but it could be a bird, it could be a finch. The host protein here is adapted to the environment. Then the environment changes. So now the host protein is maladapted to the environment. And so the host protein undergoes adaptive evolution because the protein is encoded by a gene which undergoes mutation, and just randomly some mutation will arise that makes a protein that is better adapted to this new environment. Now, at the same time, there are lots of other mutations that occur in that gene, and so over time what happens is called purifying selection. The deleterious alleles are removed. The deleterious mutations are removed. And the neutral ones stay there because they don't have any effect. And so now you have a protein in the new environment. It's maintained there. It's called positive selection. We have now positively selected for this change that makes the protein better adapted. Negative selection would be removal of the deleterious alleles. Darwin, so that's a definition of evolution to start. Darwin would have loved viruses because they're the best examples of evolution by natural selection. In other words, you have an organism, the environment changes, and you select a new crop of organisms better adapted to the environment. And the cool thing about viruses is that evolution is so rapid you can follow it in real time. You can do an experiment in a day and get evolved viruses by the end of the day. But he didn't know anything about viruses because he worked long before viruses were discovered. So we'll define viral evolution as the constant change of a viral population in the face of selection pressures. I made up the other saying, you walk down a dark alley and get in trouble. That's evolution because you don't know where you're going. You just say, oh, that's an interesting place there. Let's go down it. You could get in trouble or you could be okay. Now, modern virology has provided lots of explanations about evolution that we didn't have before. One of the reasons is because viruses reproduce so quickly, right? A cow takes a year to reproduce. If you want to study cow evolution, it's going to take you a long time. But viruses grow very quickly. And as host populations grow and adapt, viruses are selected that can infect them, and that causes new viral populations to emerge every day. It works the other way. Viruses can also be forces in the evolution of host populations, right? If the host cannot adapt to a lethal virus, the population may be exterminated. And this has no doubt happened. Both of these have happened over the years, and mostly we don't know about it. So humans can suffer greatly from virus infections, but we've never seen it, humanity being extincted, of course. But other species likely have. The public is always confronted with the reality of evolution, viral evolution, even if they don't believe in evolution. Many people do not. They do not believe in evolution. They do not believe that life arose on its own. But viruses evolve, and you can't deny it. Once someone wrote to me, said, you say this virus is 400 million years old. That's testing my beliefs. And I wrote, it doesn't matter what your beliefs are. This is the fact. This virus is 400 million years old. Just deal with it. So evolution of viruses. New viruses arise all the time. That's evolution. AIDS, West Nile disease in the US. So that virus had to adapt to be in the US. Hepatitis C virus, Ebola virus disease, Zika, COVID-19, and who knows what else. Every year, regular bouts of influenza and common cold viruses, drug-resistant viruses like HIV. That is all evolution. And you cannot deny it. Evolution happens. And in fact, virus evolution is faster than people can understand because it happens on a daily basis. It can happen on a daily basis. So first today, I want to talk about what I call the four main drivers of evolution. What properties enable viruses to evolve? And remember, evolution is not something viruses choose to do. When people say the virus evolved to do this, this is an incorrect statement. It did not. Evolution happened with mutation and natural selection. So the four main drivers, first, large numbers of progeny. You can't beat having a lot of offspring, and we'll see that often today. Large numbers of mutants. At every reproduction cycle, viruses make huge numbers of mutants. You know this already. I've told you this. Third, what we call quasi-species effects, and we haven't talked about that yet, but we'll talk about that today. And finally, selection. So the first three make lots of mutants, and then you just have to select from that pool whatever you need to be better adapted to the environment. Now, in terms of large numbers of progeny, I'll just give you two examples. Hepatitis B virus and HIV. And this is a table that shows you virus, the half-life of virus in the plasma for HBV and HIV. In other words, in 6 hours, 50% of HIV in serum turns over. 50% becomes inactivated, and that's replaced by new particles. So the daily turnover you see is 50% and 90%. Total production in the blood over 10 to the 11th for HBV, over 10 to the 9th for HIV. So these are just two examples of how virus-infected cells make large numbers of progeny. And when you have a lot of progeny, you have a lot of mutations, and you can do selection on those to get what you want to fit the environment. Now, replicating viruses make large numbers of mutant genomes. We've already talked about that when we talked about polymerases and their error frequencies. In fact, evolution can't occur without mutations. If we had no mutations, we would not evolve, and it's not likely that there'd be life on Earth. And as I told you before, RNA or DNA, both, when they're copied, they both undergo mutation, and DNA polymerases have error correction to take care of a good part of that. But If you remember one thing maybe from this course for the next 50 years, viruses are always mutating every reproduction cycle. So headlines like these are completely ridiculous. The Ebola virus is mutating. Really, why don't you say the Earth is round? That would be a comparable headline because it's so obvious. The coronavirus is mutating. What does that mean for us? Coronavirus could mutate, China officials warn. Here's the National Health Commission Vice Minister of China. The disease has been transmitted through the respiratory tract, and there is the possibility of viral mutation. There's no possibility. It is a fact. Viruses mutate at every reproduction cycle. Yet, for some reason, the press cannot get this, and most people don't understand. Only you, among few others in the world, will get this. Right? I hope you do. Don't forget it. Now, mutation rates, I've shown you this before, but let me show it to you again, roughly correlate with genome size. So here's the mutation rate in terms of substitutions per nucleotide per generation. And this is the genome size. And so these different colors are different kinds of viral genomes. Viroids have very high mutation rates here. Viroids are just a piece of RNA, single-stranded RNA. There's no way to correct errors. So that's probably why they have the highest mutation rate. And then we have RNA viruses with the next highest mutation rates. And then the DNA virus, here's double-stranded DNA viruses. They're even lower. And bacteria mutation rates, the hosts are even lower. And what's interesting is that the single-stranded DNA viruses have a higher mutation rate than the double-stranded DNA viruses because there's only one strand for single-stranded viruses, so there's no other strand to use as a template to fix the errors. Now we're going to talk about the quasi-species concept, which is an important concept for you to understand how viral evolution works. Now the name is not great, unfortunately, but we're stuck with it. So this was published first in 1978, a paper called Analysis of an RNA Bacteriophage Population, and the phage was called Q beta. And the authors of this paper said a Q beta phage population is a dynamic equilibrium with viral mutants arising at a high rate and being strongly selected against on the other. The genome is not a defined unique structure, but rather a weighted average of a large number of different sequences. Okay, and they called this a quasi-species, that viruses exist as distributions of non-identical but related replicons called quasi-species. Some people will use the term mutant swarm or mutant cloud. You can use whatever you want. I'll use quasi-species because that's what the first authors used to describe this. Now, this discovery was way ahead of its time. In other words, it meant if I gave you a tube of a virus, it's not just one sequence, it's many. How many, I'll show you in a moment. But people thought viruses in a tube all had the same sequence. And this showed that it was not the case. I was in 1978? Graduate student. Had no idea what this meant. And only many years later did I understand it. But this plays an important role in evolution, as you're going to see today. So what is a quasi-species? On the left is what the virus genome. If I hand you a tube of poliovirus, every genome has a different sequence in that tube. And that's shown here. Each of these lines is a genome, and the different symbols are mutations. There are far more differences probably, but just to give you an idea, every genome is different in this population. That's why they called it a quasi-species. Now, in 1980, I sequenced the genome of poliovirus, the first time it had been done. And I generated this sequence. And I published it. But guess what? It was a myth. This is a consensus. It's not the sequence of any one virus in the tube. that I use. It's a consensus because sequencing methods at the time and today still are not sensitive enough to sequence individual molecules. And so this is a consensus of what was in the tube. But what is actually in the tube is shown on the left. So that's why I call this the myth of genome consensus sequences. If you go to GenBank and search for any virus, you can find many genomes. People have sequenced all of them many times. But in fact, those sequences are consensus. They're an average of what was present in whatever stock the people used. So think about it this way. For a RNA virus population, the genome sequences cluster around a consensus, or you could say an average, but every genome can be different from the next genome. So, in fact, a genome with my poliovirus sequence may not exist in the population. That's just an average of all the different sequences. I'm talking about RNA viruses here. Quasispecies also occur for DNA viruses, but not to the same extent as RNA viruses. So what this means in terms of infection, so we've talked about infections of cells and of hosts up until now in this course, assuming that it's initiated by viruses with the same genome sequence. But that's not true. Virus infections are initiated by a population of genomes, not a single virus genome. If multiple viruses get into a cell, let's say a host, make it simpler, they're going to be all different genomes. And the viruses that are produced are the result of selection forces in the host, and they're going to be a slightly different quasi-species. And those viruses that can infect a new host reflect the selective forces acting on them outside of the host. So you have selection forces in the host and outside of the host. They're acting on this quasi-species. Now, quasi-species arise by mutation, but they can also be arising from recombination and reassortment. You've heard about these here before. Recombination is when a polymerase is copying one molecule and it suddenly moves to a second molecule such that the product has sequences from both molecules. That's a recombinant. Reassortment is something that happens with segmented genomes. You have two different viruses with segmented genomes. Influenza viruses shown here. They co-infect a cell. There's a influenza virus with red genomes and an influenza virus with blue genomes that co-infect the cell. So these are genetically different. All the RNAs are mixed as they reproduce in the cell. They actually reproduce in the cell nucleus, of course. And then what's packaged can be a mixture. So we have some red viruses and blue, the parental, but then we have one here that has one red segment and 7 blue segments. So that's a reassortant. So recombination and reassortment contribute to the quasi-species. Mutation, recombination, reassortment. So what this means for selection is that when a virus infects a cell or a host, it's a quasi-species. And it may be that a rare genome with a particular mutation may survive. It may be the only one that reproduces well and can move on to another host. So that mutation will be found in all the progeny genomes. But there will be other mutations from the quasi-species that just go along as passengers. They are not positive, they are not negative, they're just neutral. And so they get a free ride. We call that survival of the survivor. So the rare genome with the selected mutation, survival of the fittest. So the product of selection is a new diverse population that shares both the selected and the closely linked mutations. This kind of diversity is good, actually. It is selected for in evolution. you can make amino acid changes in viral polymerases that reduce the frequency of mutation. For example, there's one in the poliovirus polymerase, G64S, that reduces the number of errors that are made. However, those viruses do not have a selective advantage. When you do competition experiments between wild type and those mutant viruses, the wild type always wins. So the mutant viruses are less fit. And in nature, low rates of mutation are not selected for. And mutants of that sort that make fewer errors are often less pathogenic in experiments in the laboratory. This has been done for multiple viruses. It's been done for bacteria. You can make bacteria that make fewer errors. They are all less fit. The bottom line is that high mutation rates are actually selected during viral evolution. They are good. Mutations are good for virus populations and for us too. That's why we don't call them mutation. We call them single nucleotide polymorphisms, because they're simply variations. Now, when we talk about mutation, this brings us to a concept called error threshold. Okay, so the idea is mutation is great for viruses, but you can't have too much. If you have too much, the genome is going to be inactivated. So there has to be a balance, and that limit is called the error threshold. So the error threshold is simply a mutation rate. If you exceed it, infectivity is lost. If you move below the error threshold, you may not produce enough mutations to survive selection. And because here's the key, you need to make enough mutations so that if you encounter a selection event, say you find yourself in a new host, you have to have enough mutations so that one can be selected and allow you to reproduce. So if you're way below the error threshold, that's hard to do. Now, RNA viruses evolve close to the error threshold. They're living at the edge. That's why I love them. They're just really wild. Whereas DNA viruses, and that's why I worked on RNA viruses my whole career. Actually, I didn't know it when I picked an RNA virus, but it seemed cool to me. DNA viruses evolve far below the error threshold. They're taking it cautiously. And as a consequence, I mean, they're pretty successful. They infect a lot of populations, but far less than RNA viruses. So let's look at an experiment that illustrates RNA error threshold. So first, we're going to do it with a DNA virus. You infect cells in culture with a DNA virus in the presence of a base analog, like 5-azacitidine. And what happens is the polymerase will incorporate the 5-aza-C as a C. So when there's a G in the template, it'll put C in, and sometimes it will put in 5-aza-C. But then when it's copied, it templates as a T. The polymerase sees it as a T, not a C, because of the chemical modification. And therefore, you get G to A transitions, a kind of mutation. So when you do this, when you add this drug to a cell culture of DNA virus infected cells, the mutation rate goes up several orders of magnitude, 100, 1,000-fold, illustrating that the DNA viruses are existing way below their error threshold. So you have to increase it a lot for you to inactivate the virus infectivity. Now, when you do the same experiment with an RNA virus, the best you can do is increase error frequency 2 to 3-fold. At best, you can't do any better than that, and then you wipe out the virus population. So that shows we are existing at the error threshold, and that's shown on this slide here. So this is using poliovirus and an antiviral called ribavirin, which is a mutagen. It gets incorporated into the genome like 5-aza-C and causes mutations. And what we're looking at here is the specific infectivity of the genome. So here's 100%, and the number of mutations. So what they did was they increased the amount of ribavirin at specific concentrations. So each of the dots is a specific concentration. And then they sequenced the virus population to see how many mutations there were per genome. So you start with wild type, which has no mutations, right? And then you add a certain concentration of ribavirin to give you 2 mutations per genome, and you've dropped infectivity by 70% already with just 2 mutations per genome. Adding enough ribavirin to get 2 mutations per genome drops infectivity 70%. And then you add another increasing ribavirin, and you've almost lost infectivity. So very few mutations. So 7 mutations per genome, and by the time you get to 16, you've lost all infectivity. This virus is existing very close to the error threshold, because if you bump up the error frequency a few-fold, you lose infectivity. So that's the concept of error threshold. Why that's important, we're going to see in the next few slides. Now, here is going to be an example of the importance of quasispecies in virus biology. Genetic bottlenecks are what happens when you push a small number of virus particles through a selection pressure. And in this case, we're going to duplicate that by doing a plaque-to-plaque passage. We take a single RNA virus, we do a plaque assay, we pick a plaque, which means we take the virus in a plaque and we make another plaque assay from it, and we pick another plaque, and we do that over and over again. We repeat it many, many times. All right, that's the bottleneck that we're exerting. After about 20 to 30 cycles of doing this, many virus populations have hardly any infectivity. They're much less fit, we say. When a virus loses its ability to reproduce, we say it's less fit. It's lost its fitness. And why is this? The environment's constant. It's the same cells, it's the same temperature, the same medium. The only thing is that we're picking a plaque at each point, at each plaque assay. So what's going on here? Why does the fitness drop? Well, because we're restricting replication to the progeny found in a single plaque. A single plaque may have at best a few thousand viruses in it. So we're pushing the virus through a bottleneck. We're taking a few thousand viruses and doing another plaque assay with a very less diverse virus population than we started with. So we start with the parent population in this bottle, which is quite diverse, and then we subject it to a bottleneck where only a small number of virus particles can get through. And that's the bottleneck here is the plaque assay and picking a single plaque. And now the next generation is different. You can see it's got more blue viruses in it, but genetically, it's very restricted compared to the original population. And so the way to look at this problem is by a phenomenon called Muller's ratchet. Now, Muller was an evolutionary biologist who found that small asexual populations, in other words, organisms that don't have sex, accumulate deleterious mutations as they multiply, because every time a cell divides, it's simply making a clone of itself, and there's very little variation. And so he said there's an accumulation of mutations as these organisms divide, and eventually they lose fitness. Now, RNA viruses, to begin with, they're close to the error threshold, right? So what we're doing is we're making a bottleneck on the population by picking a single plaque. And as that plaque grows, mutations are accumulating. And then as we pick the next plaque, mutations are accumulating. And that's why fitness declines, because you're accumulating lots of mutations in these viruses as you go plaque to plaque. But we're not we don't have the population diversity that we would normally have if we didn't have a bottleneck. So the reason it's called Muller's ratchet is a ratchet is a kind of gear that moves forward, but it has a little lever in it so it can't go back, okay? And so each round of error-prone replication acts like a ratchet, clicking relentlessly as mutations accumulate at every replication cycle, and you can't go back because you've restricted the population size by this bottleneck of doing plaque-to-plaque passages. So let's look at it in terms of the quasi-species. So on the left is our initial population. And if we were to take the whole virus and simply infect HeLa cells, we would be drawing from the diversity of the whole quasi-species. And if we kept doing that, there would be no drop in fitness. But when we do the bottleneck experiment, we take one or a few genomes. Here, let's say this one, we pick a plaque, and now all of the viruses in the plaque look pretty similar. They don't look like the parent. And therefore, if you keep doing this, you keep picking plaques, you get accumulation of mutations and decreased fitness because you don't have the diversity that you have in the original population. So why is the diversity important? Well, here's an example before I tell you. Here are a bunch of viruses showing you fitness decline after passage through a bottleneck, which is a plaque assay, plaque-to-plaque picking. So bacteriophage, after 40 passages, 22% decrease in fitness. All that means 22% decrease in fitness is that it's growing 22% worse. VSV after 20 passages, 18%. Foot and mouth disease, 30 passages, 60%. HIV, 15 passages, 94%. And another phage, 20 passages, 17%. So this is a universal property. These are all RNA viruses, by the way. And are there bottlenecks in the real world? Yes, absolutely. So when you are infected by respiratory droplets, by virus within a respiratory droplets, there are not that many viruses in that droplet. It's a few thousand viruses at best. So it's really a bottleneck. When you have a latent virus infection, herpes simplex in peripheral ganglia. When it gets reactivated, there are a small number of particles that are made. Or if you're bitten by an insect and the insect delivers viruses, again, small numbers of viruses, thousands at best. So why isn't there a bottleneck then? Well, in our experiment, you could avoid the ratchet if instead of just passaging one plaque, if you picked multiple plaques and passaged them, then you would have greater diversity and you would avoid the bottleneck and you would avoid a fitness decline. And so more diversity in the population, the reason is now, which I had said I would tell you a few slides ago, when you have more diversity in the population, it enables the virus to make a mutation-free genome by recombination or reassortment and removing the mutations that negatively affect virus reproduction. So if you have a nice quasi-species of diversity, you can then, even if you're accumulating mutations during passage, you can remove those by reassortment or recombination. So for example, you have 2 mutant viruses that have arisen through selection through this bottleneck, and they don't grow very well. But if they could be combined with each other, they would make a recombinant with the correct mutations that would overcome this fitness defect, and reassortment can do the same thing. And so, in fact, when you get infected with a droplet or a mosquito bite, that virus re-expands within you. There's no more bottleneck within you. So it multiplies extensively, and there's a beautiful quasi-species before it moves to the next host. So there is no bottleneck in that case. Now, another example that reinforces this idea that recombination is important to rescue viruses from a bottleneck, right? That's what I've been telling you. There is a poliovirus polymerase. Remember, there is an amino acid change here, G64S, which reduces the error frequency of the polymerase. And there's also an amino acid change on the other side of the polymerase that reduces recombination. This recombination defect exacerbates ribavirin-induced error catastrophe. Remember, when you add increasing amounts of ribavirin to poliovirus, you lose infectivity rapidly. It gets lost even more rapidly if you use a virus with this amino acid change that decreases recombination. What that means is that recombination is needed to counter error catastrophe. So I'm using exceeding the error threshold as an example, but putting virus through a bottleneck is basically the same thing. So I hope that you understand what I've been trying to tell you. I've been trying to explain why quasi-species are so important, because diversity of the population is important for survival of individual members. You remove the diversity and the population as a whole suffers. That's what we see in Muller's ratchet. All right, so you know now that viruses make a lot of mutations and that they exist as a quasi-species, and the quasi-species is what infects hosts and is important for the ability to adapt to a new environment. So let's look at some examples of selection. Mutations need to arise for evolution to occur, but they have to be selected. To put it in terms of our second slide, if a protein is maladapted to the environment, you have to select the right protein out with the changes that make it better adapted to the environment. And so we're going to use genetic shift and drift as examples of selection processes. These are what occur with many viruses, including influenza virus. Now, what is going on here is that when the viruses are multiplying in the presence of antibodies or cytotoxic T cells, you will get viruses resistant to the antibodies or the CTL. That's the selection that is going on. You have a quasi-species of viruses, antibodies that neutralize, and within the quasi-species, there are some viruses that can evade the antibody or the cytotoxic T cell. So drift is diversity occurring from copying errors of the nucleic acid and immune selection. This could occur every time a genome replicates. And shift is diversity arising after recombination. So let's take a closer look at this. So influenza viruses, I don't know if you remember influenza viruses. They have envelope viruses with a hemagglutinin and neuraminidase in the membrane. And they're classified by serologic testing of the HA and the NA proteins. And we classify them by combinations of H and N, H, X, and Y. So we've discovered 18 different hemagglutinins and 11 different neuraminidases. H1 to 17 can infect birds. Also, all of them can infect birds. H1, 2, and 3 can infect and transmit between humans. They're human viruses. H5 can infect humans occasionally, very rarely, but they don't transmit well. Maybe we'll have to change this one day, but so far we don't. Okay. So antigenic drift occurs when the hemagglutinin molecule, which is shown here, undergoes amino acid changes as a consequence of mutation. And these colored areas are different epitopes in the HA to which antibodies bind. So when you get infected by influenza virus, you're going to make antibodies to these sites, and these antibodies can neutralize infectivity. But one amino acid change in one of these epitopes can cause the virus to evade antibody, and that makes us change the vaccine sometimes from year to year. So that's an example of selection by immune selection by antigenic drift. The virus is neutralized by the antibody, but within the population that's in you, there is a virus that can evade the antibody, and that one will grow, will reproduce, and transmit to other people. And that makes us change the vaccine. Same thing with SARS-CoV-2. Here's the spike protein of SARS-CoV-2 on the right. In fact, this is the spike protein bound to ACE2. ACE2 is in red. And on the right are some amino acid changes that are epitopes, are part of epitopes that are bound by neutralizing antibodies. And these amino acid changes abolish the binding of the antibodies. So as variants arose or still are arising during circulation of SARS-CoV-2, they are less able to be neutralized by the antibodies elicited by the vaccine or by monoclonal antibodies. That's why we've stopped making monoclonal antibodies against SARS-CoV-2, because it changes every 6 months and it's impossible to keep up with the production. But they try to match SARS-CoV-2 vaccines to the circulating variants when they detect them and if they are deemed to be important enough. In other words, infecting enough people. So here's an example of the emergence of Omicron. These are the share of analyzed sequences in the preceding 2 weeks that correspond to each variant group. So you can see in many countries, Delta variant was still circulating, but you can see the red, that's Omicron, is starting to emerge. It first emerged in South Africa. So you can see when this sample was taken, this is December 13th, 2021, all the isolates were already Omicron. It had a certain number of amino acid changes, many of which caused evasion of antibody. And you can see the numbers of Omicron increasing. Eventually, it spread to every country in the world. And we still have Omicron variants circulating. We haven't named it another Greek letter. All right, so that's antigenic drift. So again, what the idea is, you have quasi-species of viruses with tons and tons of mutations. And if you throw an antibody at the virus, in that quasi-species, there's going to be a mutation that leads to an amino acid change in the target of an antibody that will lead to antibody evasion. That's antigenic drift. All right, antigenic shift occurs when viruses reassort and they escape antibody as a consequence. So here are diagrams of most of the influenza pandemics that we know about. The first one, 1918, this seemed to be started by an influenza virus from ducks. So this was an H1N1 virus, and all of the genome segments are red as a consequence here. This virus seemed to go directly into people. That virus circulated in people until 1957. Now, every year it underwent antigenic drift. So the virus that was still circulating in 1956 was nothing like the 1918 virus. In 1956, another pandemic of influenza virus took place, 1957. And there the virus acquired a number of segments from an H2N2 virus from a duck. So we show these as yellow here. And this virus, 1957 H2N2, acquired 3 segments from this H2N2 virus. So the red ones are still from 1918, but you see both the HA and the NA are now from the 1957 H2N2 virus. So nobody has immunity to H2N2, right? H1N1 has been circulating for years. Suddenly, H2N2 begins to circulate. Nobody has immunity, and therefore, pandemic. Have no immunity to a virus. You introduce the virus. If it's able to transmit well, you have a pandemic. All right, so that virus circulated until 1968, and then it underwent reassortment with a another virus from ducks. And you can see the blue segments, PB1 and HA, came from the duck influenza virus. But again, we're carrying along the 1918 segments, the 1957 segments. And again, the HA being brand new, it's an H3NA. People have not seen this. They're not immune, so we have a pandemic. All right, so now in 2009, we have a pandemic caused by H1N1 virus. And this is a reassortment of many different influenza viruses. And it is here on the left are 4 different kinds of influenza viruses that contributed RNA segments to this virus. So we have Eurasian swine influenza viruses contributing 2 segments. Eurasian swine are pigs that are shipped back and forth between Europe and Asia. They're different from North American swine. We don't typically take or send pigs anywhere. Underneath is the classic swine H1N1 virus. This is the virus. So in 1918, when H1N1 went into people and caused the pandemic, it also went into pigs for the first time. And it stayed in pigs until this day. It's still in there. Many pigs are infected with H1N1 influenza virus that originated from 1918, but it's drifted for all those years. And so in 2009, it had drifted long enough so that nobody had immunity to the H1 segment. So 3 of those segments went into the 2009 virus. Some segments came from the H3N2 virus, and some segments came from an avian influenza virus. And these 4 viruses were circulating as far back as 1990. In 2000, we could detect 2 intermediate reassortants circulating in North American swine, and then 2009, the final reassortant emerged. And this is thought to have emerged. So the final virus emerged in a pig, on a pig farm, and then that went into humans, which then triggered the pandemic because the H1 was completely different from what people had seen before. Now today, both this H1N1 and H3N2 virus are circulating, so we vaccinate against both of them. But this shows you how just swapping segments gives you a brand new hemagglutinin to which we have no immunity, and that causes a pandemic. So presumably it's the antibody pressure on the virus population that does this. Okay, now to end up, we're going to talk about Alice in Wonderland. This is the Red Queen conflict that I referred to, or host virus arms races. So basically the idea is we have a virus that infects a host, and there's some host protein that can block virus infection. Of course, since viruses mutate so often, eventually a virus will be selected that cannot be blocked by the host protein. In response, the host changes. It takes longer for the host to change, but the host will change so that it can block the virus again. And this goes back and forth for tens of thousands of years. So that's what we call a host virus arms race, or a Red Queen conflict, because the queen said, here's the Red Queen, and here's Alice. She said, around here, it takes all the running you can do just to stay in the same place. So the virus and the host are both running, and nobody's gaining an advantage. It's a classic Red Queen conflict. Now, we can actually study these Red Queen conflicts. And let me show you how we do that. So let's say we're interested in host virus conflicts in primates. So the first thing you do is you assemble a phylogenetic tree of all the different primate species that you're interested in and how they're related to one. And we have genome sequences of all of them. And what you do is you subject those genome sequences to DNDS analysis. So what's that? So as you may know that when codons change, they can have synonymous or non-synonymous changes because the amino acid code is degenerate, right? Some amino acids can be coded for by up to 6 codons. A synonymous change, and the word synonym means the same. Synonymous means that mutation doesn't change the amino acid. Non-synonymous means it changes the amino acid. It's no longer a synonym. So what you do is use a computer program to scan the sequences of all these primates, and you look for the DNDS ratio. And if it exceeds 1, that's evidence that there have been a lot of non-synonymous changes, and you can actually identify synonymous and non-synonymous changes among these ortholog sequences. And then you can identify lineages within this phylogeny that are positively selected, in other words, that stay throughout evolution through these branches of the tree. And you could look at the codons and say, looks like there's evidence for positive selection at this codon. And then you can experimentally address this. You can have cells from the primate. You can have viruses that you engineer the changes in, and you can say, yeah, this virus change neutralizes the host change, and so on. So that's how you do it. And so here's an example of the results, 2 examples of the results. These are 2 host defense proteins, TRIM5 alpha and PKR, protein kinase R, that have been involved in host virus conflicts for many, many, many thousands of years. And if you look at TRIM5 alpha, there are these red arrowheads are positively selected amino acids that we've identified by the experiment we just did. And you can show that those amino acids in TRIM5 interact with the HIV capsid. And so TRIM5 and HIV capsid vary at these positions back and forth, but these are the changes in the TRIM5 alpha. PKR, on the other hand, blocks many different viruses, and so it has signatures of conflict with herpes viruses, influenza viruses, pox viruses, Hep C, pox, frog pox virus, and HIV throughout the RNA binding domain and the kinase domains. In other words, these are positively selected amino acids, which one amino acid will block virus inactivation and the other will not. The way you can look at it is that here's the host defense protein, and it has multiple surfaces that can bind viral proteins, and these viral proteins can change as the host changes as well. So these are the changes of the host to evade the viral counter defense proteins. So that's why it's a red queen conflict. Nobody wins. The host is still around and the virus is still around. Now, I've told you today that there is a lot of viral genome diversity, yet there are only 3 serotypes of poliovirus, yet related viruses, rhinoviruses, there are over 150. There's 1 measles serotype. We use the same measles vaccine as we have since 1960. There's no drift to evade measles vaccine. Yet influenza virus varies antigenically continuously. Now, what we have learned from various studies is that poliovirus and measles virus can undergo antigenic change, but they rarely do so in humans. Apparently, there are structural constraints in some viruses, poliovirus and measles virus, that limit the amino acid changes that can be sustained in the proteins that interact with the immune system. Another selection force, so we've talked about immune selection in terms of shift and drift. Another selection force is virulence. is virulence positively or negatively selected? In other words, if it's a positive trait, increased virulence is selected for, maybe because higher fitness gives you higher viral loads and that causes more disease. But it could be a negative trait where increased virulence reduces transmission between hosts because they die faster and reduce exposure to unexpected hosts. Or it could be neutral. Maybe increased or decreased virulence is a consequence of a different selected phenotype that happens to impact on virulence. We don't know which one of these is correct. I think all three are likely, but there are many avirulent and virulent viruses out there. So it's hard to know what's going on. Now, you can't do experiments in humans to understand virulence and evolution of virulence, but there is a very famous experiment in the evolution of virulence, which was done in Australia. So in 1859, the European rabbit was introduced to Australia for hunting. So Australia had its own rabbits, but the hunters couldn't catch them. They couldn't shoot them because they were too fast. So they brought the European rabbit in, which was slower and they could shoot them, but there were no predators of the European rabbit. So within a short period of time, it reproduced to plague proportions, and all the waterholes were surrounded by rabbits who reproduce really quickly. Look at them all. So Australia had a rabbit problem. They didn't know what to do with it. So they decided to release a virus to kill the rabbits. So myxoma virus was released in the 1950s to rid the continent of rabbits. So the natural host of myxoma virus is the cottontail rabbit, one of these two, I don't know which one. The virus is spread from rabbit to rabbit by mosquitoes. They get warts, and they're fine. They're not a problem. But the European rabbits are a different species. One of these is a European rabbit. In them, myxoma virus is 90 to 99% fatal, right? So the Australians thought, we're going to wipe out all the rabbits and we won't have this problem. Do you think it worked? I wouldn't be telling you if it worked, right? So they released the virus the first year, 99.8% mortality rate in the rabbits. But those 0.2% of rabbits, did they multiply? The next year, the mortality rate dropped to 30%. So the rate of killing was lower than the rabbits' reproductive rate, and they could not eradicate the rabbits with this virus. It turns out that both rabbits and viruses make a lot of offspring, right? And so you have a nice mutant pool to select from. Viral variants were selected that kill fewer rabbits so that rabbits could overwinter and spread the virus in the spring via mosquitoes. So instead of killing the rabbits, they survive to the next season and they can spread. So the reduction in virulence may reflect that. The rabbits also have changed. Many years later, the genome of these surviving European rabbits in Australia were sequenced and many immune system gene mutations were identified. And we think those changes make the rabbits more tolerant or resistant to the virus. So this is one outcome that you might predict in the evolution of a host and the virus, right? Initially, when the virus first gets into the host, there's massive killing, but if the species is highly reproductive, the mutations in the host and the virus may lead to an equilibrium. But what about people? We can't do virulence experiments in people, but we know a few things and give us some clues. So Lassa virus, Ebola virus, HIV-1. These are all viruses that entered people from animals. They tend to be virulent. HIV is still as virulent as when it first jumped into people in the 1920s. But there are some viruses that are not as virulent. They don't kill you. Measles, poliovirus, well, they can kill you, but less so. So what happened? We don't know because we don't have the original poliovirus that jumped into people. We don't have the original measles virus that jumped into people. If we did, we might be able to compare their genomes. We would like to be able to compare the human genome as well, today versus when the virus jumped in, but we don't have that data. Now, SARS-CoV-2 jumped into people in 2019 in November, December. And so that's one we could possibly track in terms of evolution of virulence. But as I told you, it's hard to measure virulence by observational studies where you just say, this person came to the hospital and they had this variant of SARS-CoV-2. So that's, and I showed you some criticisms of that before. But it's still possible that as SARS-CoV-2 continues to evolve, we'll see changes in its virulence. For example, and these changes are likely to be mediated by population immunity. There are 5 common cold coronaviruses which circulate continuously among people and cause mild infections. These originally came from animals many years ago and probably caused pandemics, although we don't have good records of it. Today, the viruses cause mild upper respiratory disease. And so some people predict that eventually SARS-CoV-2 will be mild like that because the problem with the pandemic was there was no immunity in the population, so the virus infected both the young and the old. And the old, being very susceptible, died off at higher rates, as I have shown you, than young people. The mortality rate in young people is lower, and so what will happen at some point is that the older population will all be immune to SARS-CoV-2 from infection or vaccination, and then the people who get infected who are not immune will be young, and they will have less disease because they're young. So that may be how some human viruses evolve, to have their virulence modulated by population immunity. Now, where did viruses come from? Well, the oldest viral stocks we have are 1918 influenza virus and Pandora virus, which there's a 48,000-year-old isolate from Siberian permafrost. They pull out ice cores and they can recover virus from them. But that's not very old, of course. But we have modern techniques that allow us to judge the age of viruses. For example, we use a technique called molecular clocks. You can calculate the mutation rate of the virus and match it with the host over time. And when we do that, you can say that the 3 major groups of herpes viruses arose between 180 and 220 million years ago, when the dinosaurs were walking the earth. So maybe dinosaurs had herpes cold sores, but the viruses were certainly there. So 180 to 220 million years ago, Jurassic Park, there you go. Okay, another thing we can do, and I think this is brilliant, is use endogenous viruses to calculate the age of viruses. Now, an endogenous viral element is just a piece of any virus genome that went into the cell genome at some point. It could be a retrovirus, but it could be just a piece of another virus that went in accidentally. So here we have 3 kinds of rabbits, 3 species, A, B, and C. And species A and B have an endogenous viral element in them. And you can sequence the genome of these rabbits and see exactly where the integrated EV is, and the species C doesn't have it. And then you do a phylogenetic tree, which says that the ancestral host here must not have had it because this species C is derived from it. And these 2 branches split about X million years ago. The EVE went in here and hence was given to both species in the same chromosomal location. So from this, you can get a maximum or minimum age of the endogenous viral element, and that tells you when the viruses were circulating. So by that, we can look at single-stranded DNA virus integrations in different species and look at them 30 million and 60 million years ago. These viruses were circulating and infecting those hosts. Very cool. But where did viruses actually come from? Where did they start? There was a pre-cellular stage of life on the planet before cells, but it was occupied by replicators. These were probably RNA-based, and they were molecules that could replicate on their own. Eventually, cells arose, and that's a big gap there. And we think that these replicators invaded cells and picked up capsid genes from the cells and became bona fide viruses. So the replicators went into cells and became viruses. And then as the cells evolved, these are protocells, very primordial cells, the viruses evolved with them. So evolution of modern cells and continuous emergence of new viruses and diversification. So there's a general idea of how we think viruses have emerged. Why don't we have new viruses emerging today? Let's make an assumption that new viruses can only arise from viruses that are here, not brand new de novo, all right? So what are all the possible mutations of a virus genome that could lead to that? Well, some studies have shown that you can mutate over half of the nucleotides in some RNA virus genomes, over half, okay? So if you have a 10,000 base RNA virus genome, you can mutate half of the bases. That means 4 to the 5,000 sequences are possible. 4 to the 5,000. And that number goes up by deletions and recombinations and reassortment. To show you how big it is, there are 4 to the 135 atoms in the visible universe, what we can see. 4 to 5,000 is huge. So why don't we have new viruses emerging? Because we can still see a flu virus is a flu virus, a herpes virus is a herpes virus. There must be some way that stability is maintained. And yes, there are many ways that stability is maintained. So that extreme alterations in the viral genome, some of those 4 to the 5,000 mutations, they will not survive because the genome, for example, is a constant. DNA can't change to RNA or vice versa. You have replication proteins, you have replication signals in genomes, you have mRNA signals, you have RNA structure and codon usage. The physical nature of the capsid, it will only hold so much nucleic acid, so it can't increase. And selection during infection. A mutant that arises that kills cells very quickly will kill the host and have the same fate as one that doesn't replicate. So basically, there are lots of constraints on virus evolution that prevent those 4 to 5,000 mutations from giving you something completely different. And let me leave you with food for thought. So we as humans are 98% chimp. Our genome is 98% homologous to that of chimpanzees. That took 8 million years of evolution to get 2% difference in our genome. A poliovirus infects you, it goes in your mouth, through your intestinal tract. You excrete it 5 days later. It undergoes the same change in those 5 days as chimps to humans. 2% of the genome changes in those 5 days. So imagine what a virus could do with 8 million years of evolution. All right, now you have the tools to understand how new viruses emerge from animals into people, which we'll talk about next time.